All right, can you see my slides?
Yes.
We're good to go. Well, it's really an honor to be able to talk about what we have been, how we've been using non-human primates in the HIV care space. But before I start, I want to first of all really dedicate this talk to all the dedicated animal care staff who ensure that the health and well-being of research animals. I mean, to be honest, I think they are unsung heroes. They make sure that these studies are done safely in a humane way and that they're really reliable. And so I really want to acknowledge them because I think sometimes they get left out in this in all the controversies. And as far as I know, most of the animal care staff that I've encountered, they're just dedicated professionals and they really take care of animals. Okay, as I said, just really dedicated to the animal care staff who take care of primates. All right, so infectious diseases, it's amazing that infectious disease outbreaks are becoming more and more common, and they seem to be increasing worldwide. We all experienced this with the COVID pandemic, but there are other infections that are also seem to be spreading, and we in the news now we heard about mpox, which spread recently, and now we're hearing about these new cases of measles. I suspect that we're going to have more and more of these outbreaks in the recent years. And so it's research in animals have really helped us understand how these infections spread. They also have helped us to develop vaccines very quickly and for treatments. I also remember a couple of years ago when there was a massive Ebola outbreak, non-human primates were really essential in developing a vaccine for that is now available for the most part for Ebola, which is a really deadly disease. So for many years, non-human primates and animals research in general have been really important for developing vaccines. And a good example was a COVID vaccine. This was an accelerated vaccine pipeline, which was really quite remarkable. But a lot of these studies was really done in animals, especially primates, to show how efficacious those vaccines can be. And so these monkey studies were really used to develop vaccines and treatments. And not only that, monkey studies are really ongoing now to help us understand long COVID. And I don't know if you have met anybody who has long COVID, but it's really a mystery disease. We don't really know. Everybody seems to have different experiences with long COVID. And really trying to understand the biology of it is really challenging. And I know there are ongoing studies now to try and understand long COVID. And so this approach to using primates to guide studies in infectious diseases is really helping us in the area of HIV, not just the cure, which we're going to talk about today, but in addition, prevention and developing new treatments. So why do we use monkeys? Well, for the most part, monkeys are biologically very similar to humans. And I want to start this by saying that, I've had a lot of criticism that monkeys are not the same as humans. And I think for those of us in the field, we know that monkeys are not the same. But there are a lot of similarities between monkeys and humans. A lot of the studies that are done with in HIV cure research, we use this species called rhesus macaques, which is Macaca mulatta. They're either of Indian origin. They're mostly of Asian origin, either Indian or Chinese origin macaques. But there are some other species of macaques that are also used very frequently. These are called pigtail macaques, some homologous macaques, and African green monkeys. And African green monkeys are what we refer to as natural hosts, which I'll get to in a bit. But, I say they're biologically similar because when we look at their immune system is very similar to the human immune system. So the human immune system is very diverse and complex. Our T cell biology is made up of cells that are naive, so cells within your body that have never are there primed to attack an infection. Humans have naive cells. Monkeys have naive cells. Monkeys have memory cells. So when you get a vaccine, what we do is to try and educate the immune system to remember that infection. And so that memory of that infection is in the compartment of your T cells called memory T cells. And monkeys have memory T cells. Humans have memory T cells. So we can give monkeys a vaccine and say, hey, remember how to fight this infection. And that education or that memory is going to live in the immune compartment. And how it lives in the immune compartment is very similar to what happens when humans are given a vaccine. So there are many similarities. The organ functions are very similar between monkeys and humans, from the reproductive system to the cardiovascular system. So there's so many different similarities between them. And with all these similarities, it means that we can use them as a model to understand disease processes. Okay, and I just give an example of this. Yellow fever is a really great example of this, of how the yellow fever virus, which kind of can infect the liver and really cause disease and inflammation in the liver. Monkey studies were really the first to show how the virus can damage the body, and can cause liver infection. And then it was even in monkey studies that these treatments, that the early vaccine was developed for yellow fever. And that vaccine is still used till today to treat and prevent yellow fever. So there is a long history of how monkeys have really been used to develop treatments, vaccines, for infectious diseases. So it's not unique to HIV. And I think we're actually quite fortunate in HIV because the animal model for HIV, which we use in non-human primates, is really a model that really recapitulates most of what we see with HIV in humans. Now, just to give you a very brief background of why this, and this slide is a little bit complex, but that's probably, I can promise, my most complex slide. But most HIVs and SIVs that cause AIDS in monkeys really originated from SIVs that are in monkeys that come from Africa. So there are these simian immunodeficiency viruses. They're currently endemic in some African monkey species. And in those species in particular, they don't really cause animals, those monkeys to have a disease like HIV or AIDS that does in people and that we see in people. Probably through millions of years of evolution, these animals have learned to adapt to this virus. And I would just highlight that there are many viruses that we as humans live with on a day-to-day basis that don't always cause us disease. If your immune system is competent, there are viruses like cytomegalovirus, Epstein-Barr virus. We live with them. We are naturally infected with them, but we've kind of evolved to kind of, live with these viruses. But there are certain viruses that come, and when we are exposed to them, they cause disease and they become pathogenic. So SIVs, we believe, were originally transmitted from some African species into gorillas, and into chimpanzees. And from chimpanzees, we believe that some of the origins of HIV really came from viruses that have passed from some African monkey species into gorillas and into chimpanzees, and then found their way into humans. And this cross-species transmission into humans, and humans had not adapted to this lentivirus. So once that transmission event occurs, then this virus becomes highly pathogenic in humans. Our immune system cannot really cope with this virus. And that's why we get the virus now that we refer to as HIV. Interestingly enough, we can also get these viruses that come from these African species and put them into monkey species that don't come from Africa, in particular Asian macaques, and they cause a disease that is very similar to HIV, what HIV does in people. And I just highlight some of those similarities here. When HIV goes into people and SIV goes into Asian macaques, the virus levels rise very quickly and they remain very high. The virus constantly changes, so we call this mutations, and it escapes immune pressure. So the immune system tries to fight it, but because it rapidly changes, the virus escapes. SIV and HIV, usually the immune system cannot control these viruses. And critically, the viruses target the same type of cells, in particular CD4 T cells. So in humans and in monkeys, you get a depletion of your critical immune compartment. In particular, we call them CD4 T cells, and these cells are gradually lost, especially in the gut. And so very early studies show that HIV, one of the first things that happens is that a lot of the T cells in the gut, CD4 T cells in particular, get depleted. And later on, studies in macaques also showed that this was the same phenomenon. In addition to this, without treatment, you have ongoing immune system activation. So your immune system always is at the state that it needs to fight this virus, and this is very similar across the two species. And finally, damage to the gut. As I said, it depletes all the cells in the gut. It damages the gut. And when it damages the gut, we have what we call the microbial translocation, which is really your gut is leaky. Bacteria that are naturally in your gut system begin to leak into the bloodstream, and it increases levels of inflammation. And this mechanism in monkeys is very similar to what we see in humans. So with all these similarities, you can see that we see this as a really good model to understand HIV, how HIV causes disease in people, how to treat HIV in people. We also have to understand how HIV persists when people are on antiretroviral therapy. And as we are really involved in, how can we come up with curative interventions? Because this is a model system that represents what we see in people. Just this slide just highlighting that when HIV remains untreated and people progress to end-stage disease or AIDS, some of the diseases that they are susceptible to because their immune system is completely destroyed.
Afam, we have lost you.
Yeah, okay, sorry, it went on auto mute. I was just saying that one of the things we see is that when people who are living with HIV go untreated and they progress to disease, some of the diseases they're susceptible to are very similar to the diseases we see in monkeys. And so that's why we continue to say this is a very good model, because when your immune system is healthy, some of these infections are very uncommon and only come up when people get to disease. But we can show that this is very similar in monkeys, where they are susceptible to pneumonia, candida, and even lymphomas caused by some gamma herpes viruses. So all this to say that we have a really nice model to understand HIV. Now, non-human primates were really important very early on to understand how to develop treatments for HIV. And if you can remember back in the day, and this is actually a slide that I got from Nancy back in the day, Nancy Haywood, our former director. But just to highlight that, very early on, PMPA was really the only drug, was the first one that came up. And to it was the first one that came up, but this drug was already available, but it was also very toxic. So unfortunately, it was a lifesaver at the time, but also it came with a lot of toxicities. However, new medications started coming up, and some early studies that really showed efficacy in macaques showed that there's another orally bioavailable drug called TDF, or tenofovir difoxyfumate, that showed that in monkeys it could suppress virus, and that actually led to clinical trials in people starting in the late 90s. And after multiple clinical trials, the FDA approved tenofovir as an additional drug to treat HIV. And since then, we've transitioned from monotherapy to now and triple therapy, and now we have so many wonderful and amazing drugs that can suppress HIV now. And we know that HIV treatment works, and it can suppress the virus. And if you're completely undetectable, or if you're undetectable and fully suppressed, then you can't transmit the virus. So it's amazing how we've really progressed. And along this developmental pipeline, non-human primate studies have been essential for testing these to show efficacy, to show safety before you move into the clinic. And I just highlight right now that the whole field is galvanized around this idea of long-acting antivirals, and the idea that we have lenacapavir now that can actually prevent infection. And it's really amazing. But just to remind everybody that Gilead Sciences that developed this drug and is now really published back in 2023, their first study showing that not only was this drug safe, but it can prevent SIV infection in non-human primates. And this was done by Gilead back in 2023. And then in 2024, Science now said this was breakthrough of the year. This is a long-acting antiviral that you just need twice yearly injection. But initial studies were done in primates to show safety. Before they moved it into the clinic. And from then, after the primate studies, this actually now led to the big study that was published in the New England Journal of Medicine, showing that what they had seen in primates was true, that they can give this injection to people twice yearly, and you can get almost a 99% efficacy or prevention of viral infection. So in some circumstances, monkey studies don't lie. They are there to test and to prove a concept, and then you can take that into the clinic. You've proven that the drug is safe. You've proven that the drug does what you think it will do, and you can then move to the clinic and then test and see whether this works. And it's an amazing story that now we have a drug that you can give a twice-yearly injection, and it really is highly efficacious in preventing HIV, and it has really galvanized the field. But I also want to highlight the contribution that non-human primate studies had played in this amazing feat that Julien has brought up. So let's focus a little bit on HIV cure. So how can we use non-human primate studies to find a cure? And I'm not going to go into specific studies because Julien is going to talk more about some of the studies that are ongoing. But just to remind everybody, as you all will know, that when people once people start treatment with HIV, the virus is fully suppressed. It doesn't actually eliminate the virus. And the problem is that there's always a small number of cells, HIV-infected cells, that remain even though people are on highly effective treatment. So you can take your pills every day, and you can take it for many, many years, but we have this pool of cells that we can't get rid of. If treatment is ever stopped, the virus can wake up from this little pool of cells. And so the challenge for us in the field, as we're looking for a cure, is to find these cells and figure out how to eliminate them. And it's really key, either to completely eliminate them, or even if we can't eliminate all of them, to have the immune system potent enough so that it can control the infection, so it doesn't cause disease. So that's really the challenge. But I think part of this complication is that these cells that we're looking at, this little pool of cells, they are not only in the blood, they are everywhere. I mean, if you think about how the immune system works, the idea of the immune system is to be able to interact and challenge, they're almost like policemen of the body. They're supposed to surveil every part of the body and looking for any potential threats. And so they're not only located in one site, they're located in many sites, because infectious agents, cancer, and all these other things can happen anywhere within the body. So you can have these cells in the gut, you can have them in the brain, you can have these cells in the liver, the spleen. They're all over the place. And so it's really a challenge to understand how we can cure, and if we can cure HIV if we don't really know where they are, or we don't have access to all these multiple different sites, or to show that whatever treatment we've developed it should be efficacious in all these different sites and all these different compartments. And so these cells can be everywhere. And so that's why having a model to be able to ask experimental questions that allows us to look in these different compartments is really essential in how we're trying to find a cure for HIV. But in addition to this, I also want to highlight that when we think about HIV and cure, we know that living with HIV for a long time also seems to have increasing effects on health. And so we have this idea that it increases risk for other health conditions. And these health conditions are really associated with what we refer to systemic diseases, somehow partly driven by inflammation. These diseases are not just diseases you can quickly treat in the blood. We're seeing increase in incidence, same things that you would normally impact when people get older. We see this accelerated increase. So understanding from the systemic level how living with this virus for a long time impacts all these different compartments. And so if you have a treatment and you want to see how this treatment impacts all these different compartments, from your social cognitive behavior, from your liver disease, kidneys, cardiovascular, all these things that we see increasing in incidence in people living with HIV, I think it's nice to look at this on the whole organism level. And this one of the papers I thought really didn't get a lot of attention at the time was an article from the New Yorker that came out many years ago, really looking at this idea of the hidden crisis of HIV, where there's an increased incidence of aging, or people living with HIV aging faster. And I thought it was really intriguing article, and I think about it all the time as we're trying to find a cure, because I think it really matters. We have really good treatment, but we also want to make sure that people living with HIV are aging healthy, and looking for treatments that allow them to age healthy is really important in my mind. So how do non-human primate models help with this? Well, they help because they can allow us to look at these whole organ systems. We can look at the brain and the nervous system, things that are very challenging to do in the clinic. We can look at the heart and the blood vessels. We can look at metabolism, how we consume food and energy and weight and all the things that interact with the body to drive or reduce incidence of diseases. We can look at the reproductive health and pregnancy. Living with HIV, we know now that children whose mothers were living with HIV, even though when the children are born, even those children do not have HIV, they are actually seem to be susceptible to certain diseases. It's really interesting. They refer to as HIV exposed but uninfected infants. And when you look at their life compared to infants whose mothers were not living with HIV, there are differences. It's really quite remarkable. And really understanding this from a whole organismal level, I think is really important. Inflammation and aging, how do these two interact in the system? And overall looking at the immune system. So just to give you a picture of how non-human primate models, not just looking at them in vitro or in a new approach methods, but how the whole organism, how we can understand the disease from a whole organism perspective. And I think that's really some of the strengths of the model. Remember, I'm always saying it's a model because we truly understand that monkeys are not humans, but we use them as a model to understand these different complex disease processes and how HIV interacts with all these complex disease processes that are very challenging to do in mice, also very challenging to do in vitro, and also challenging to do in people. So what are the advantages of non-human primate models? Just to say that they offer us this opportunity to design experiments in a way that is very controlled, because there's a lot of variability and heterogeneity in studies. Everybody's not the same. And when you want to ask an experimental question, when you have so much variability, it's hard to find an answer. So sometimes we want to be able to control everything, control the timing we give infection, whether we infect the animals early or late. It can model what happens to people who acquire the virus early and are treated early versus people who acquire the virus late and actually are treated much later in the course of their disease. So we can actually model this in the non-human primate model. We can challenge them either through mucosally, which is similar to what happens most vertical transmission with HIV, or we can actually challenge them intravenously, which is very similar to what happens with injection drug users and how they get exposed to the virus. So we can actually control all these different variables. We can give them different viruses and different viral strains depending on the question we want to ask. But most importantly, I think, is the idea that we can actually look across the whole organism. And actually, in my lab, we have studies we're trying to look and understand HIV in the brain and understand how certain immune interventions affect the ability of HIV to persist in the brain. We're also doing some PET imaging studies to actually see, in collaboration with Timothy Hendrich at UCSF, who is actually doing PET imaging in people living with HIV. We are doing similar studies in non-human primates because in his case, the disadvantage is that even if he sees signals, he cannot just go in and biopsy and do surgeries on those patients because ethically is challenging. But for us, we have the opportunity to do such. If we see signals, we can go in and do PET-guided surgical biopsies to understand what is that signal and why is the virus there relative to other sites. And so really, that's a huge advantage of the non-human primate model, to be able to look at these different sites and do things that are very difficult to do in people. So not only to understand the biology of HIV, but also to understand how the different treatment interventions that we hope we can translate to the clinic impact the virus, or in fact how the virus persists in different tissue compartments. Now, I say all these things, but I also want to highlight that there are also challenges with non-human primate research. But first of all, I want to highlight the fact is that it is actually very expensive. And so it requires a lot of investment. Because you can imagine there's a lot of regulatory things. Animals need to be taken care of. They need to be bred. They need to be housed. You need to provide feeding. You need to train highly qualified staff to be able to look out for them. You need excellent veterinary care. which we have across all the all the primate centers in the country. And so all that, the infrastructure to do non-human primate study is quite expensive. And actually, we're quite excited that that Congress actually gave the non-human primate centers an extra 30 million in the last appropriation to actually continue investing in infrastructure in the non-human primates, in the national primate centers. Now, also, I mean, just like in humans, there's a lot of genetic variability. So genetically, monkeys are not always the same as people. There are certain genes that are expressed differently. And so we have to understand the genetic diversity or differences between monkeys and people when we're interpreting our results. There's also the idea of biosafety and containment. I mean, it's all very complex. As I said, we have to have huge infrastructures that actually have to be for housing and animals. We have to make sure that people are safe who are working with animals. So there's a lot of logistics that goes into that. And then we also have to say that, well, because we see it work in monkeys, do we always think it will work in people? And that is the challenge with translational studies. For the most part, we design experiments that we believe should have appropriate translational potential. But we also have those caveats. We won't know 100% if we see something that cures a monkey until that has gone into the clinic. But we can try as much as we possibly can to ensure that when we look at this treatment and we do all the safety and toxicity testing we can in the primate model, we will have a high degree of confidence that potentially if that goes into the clinic, it should be safe. But we can't say that for 100%. And I think for those of us in this field, we understand these limitations. There's also regulatory constraints with non-human primate studies. We are highly regulated. There's so many levels of regulation from our institutional ICOC review board to nationally, the Food and Drug Administration. We are under so many different guidelines, and we have to have a lot of these things in place because we can get in trouble if we don't follow them. So, I mean, I always want people to realize that we don't just do monkey studies for the sake of it. This is a very highly regulated field. We have to have multiple levels of approval before we can proceed with a study. Even when we are proceeding with a study, we still have to have oversight over the study. If anything goes wrong, we have to self-report immediately or we get into trouble. So there's so many things. Everything is highly documented and it's reviewed and inspected. And we have many levels of inspection, not only just as a PI individually, but also as an institute, as a center. We have so many levels of regulation. So it is probably the most tightly regulated area of research you can ever possibly do, apart from probably if you're working with bioterrorism type of things. There's also some limitation in reagents because sometimes the reagents we use, they don't always work as they work in monkeys. They may not work the same in people. And as I said, monkeys aren't always.
Tell us what reagents are.
Oh, sorry. So, when I mean reagents, for instance, if we want to analyze things like inflammation, if we want to analyze how the immune system is operating, we rely on things that we need to buy from companies. For instance, we need to buy antibodies to look at how one immune cell is changing over time. In some situations, we have all these antibodies that seem to work the same, but sometimes they don't work the same, or we have to get different clones of antibodies, or we have to send sample off for analysis. Most of the big analysis platforms were developed for humans. And sometimes, if we want to understand levels of inflammation, we could send the blood off to a company to measure levels of inflammation. But they develop that for people. They don't necessarily develop it for monkeys. And so we always have to know, okay, in some cases it's fine, but some cases it's not always accurate. So we have to understand what the limitations are. What is the assay really telling us? And is it exactly the same? That answers the question. And just to highlight, as I said, monkeys are not perfect. And sometimes, it can take a long time to move from a monkey to a human because it takes, there are gaps. You can't just quickly do an experiment and then tomorrow design a clinical trial. And so this is a long process. So just to summarize, the challenge of the non-human primate studies is that it's expensive. most of the time we don't do a lot of monkeys because it's half of the cost. And so, we might do a 30 monkey experiment, but we can't know if in 30 monkeys what we've tested is going to be the same in 1,000 people. But these are preclinical studies that give us early evidence of things we might want to take to the clinic. We only use monkeys when possible, so we don't just think about it. We have to design experiments that are really relevant, and we don't just come up with studies just because. And so non-human primate studies are really reserved for the most important questions. And I'm just going to end very quickly on this idea of ethics, because, as many of you may have known, there's a lot of pressure on non-human primate studies now. In fact, the FDA came out with this announcement that they're not going to require animal studies for preclinical toxicity testing anymore, especially for drugs like antibodies, which is fine. But I just want to highlight and remind everybody that there was something called the Nuremberg Code, which was established in 1947, and that was following the doctor's trial after the horrific experiments that were carried out during World War II. And this code set important international standards for the ethical medical research, and including one of the main principles is that experiments with humans should only be supported by prior studies in animals. We don't want to be rushing to put anything into people that we are not sure is 100% safe. And so that was really, this code came up back after World War II, and it has stood the test of time. I just want to remind everybody, and I think it's really critical that especially when thinking about a cure, and that these are not interventions that might be trivial. It's really important for us to make sure that we're not rushing to put things in people that we don't think is safe. And I just want to highlight too that even though the FDA is not going to require this, but the pharmaceutical companies and biotech companies who are developing these products are legally obligated to make sure that whatever they put in people is safe. And I suspect that they will continue testing these things in animal models as part of this responsible drug development before that goes into people. And then also to say that even though the FDA is saying this doesn't mean that there is a general consensus that non-human primate studies are no more relevant for HIV cure research. And in fact, I've had many interactions with program officers and people at the NIH, and I think they still strong believe that in the area of HIV cure, non-human primate studies are going to play an essential role going forward. So just to highlight, we take this very seriously. We understand that the limits of monkey studies and that they're not perfect. We work hard to use as few animals as possible. We're actively testing new research methods so that we can replace the use of monkeys for certain questions. And if these better alternatives come available, we think that naturally we will see a decline in the use of these animal models, but we are not there yet. And I think it's going to take time for us to get there, where we can say that, yes, this intervention, we can test this in a different system and we necessarily don't have to use an animal. But we are working towards that, as most people are. And I just to highlight too that the non-human primates that are used for research, they are mostly purpose-bred. They don't harm wild population of animals. These animals are all here and they're purpose-bred, and so it has no disruption to the wild animal population. And I'm just going to end by saying thank you and for this opportunity. Thanks for listening. We have a lot of animals on our campus, but also to say that our campus also has all these wonderful creatures that come out during the spring and they really brighten our day. And I will stop there. Thank you.
Thank you, Afam. That was wonderful. I just would like to reiterate too, our country has a history of doing experiments on prisoners and on people in the service. So, if we don't have animal research, somebody's going to have to get in line to be the first to try these things. I hate to think of this, but I wonder if that might be people in ICE facilities too, trapped there and just at the mercy of the powers that be. So, I mean, without the animal studies, studies would have to be done first in people. And we've been very lucky in HIV that our drugs haven't done haven't killed anybody. All right, I mean, just be straightforward. But that's not the case in all areas of research. I mean, we've had toxicity deaths in FIAU, in liver disease. We've had that gene therapy experiment. That experiment that wreak so much havoc on one person set that area of research back years. So the FDA is talking about saving time, but if somebody dies from one of these compounds or interventions, that research is going to suffer as well, way longer than the 3 months that they say they're saving by looking at cells in petri dishes rather than in live animals and in live organ systems. So
we're delighted to have Julien Clain who is a postdoc researcher from the Emory National Primate Research Center in Atlanta, and he works with Mirko Perardini, who couldn't be here today, but they are researchers in both the ERACE and RID-HIV Collaboratories doing cure research. So we're delighted to have him talk about some of the research that they're doing that include the NHP model. So Julien, thank you. Take it away. You're still muted.
I'd like to thank Dr. Paul Johnson for joining us too, who's the director of this section of the Primate Research Center at Emory. So thank you for joining us, Dr. Johnson.
Okay, do you hear me now?
Yes, perfect. Looks good. Sounds perfect. So go ahead.
Okay, great. So I was telling good morning or good afternoon, depending on where you are. So thank you, Jeff, for the introduction, and thank you to the organizers for the invitation. It's a real pleasure to be here online with you today for the first time. And I'm also glad to have the opportunity to share our work, as I think it's essential that both scientists and the community remain closely connected. This dialogue, I think it's a key to ensuring that our research stays meaningful, relevant, and also impactful, right? So today I will be presenting 2 studies that we are currently conducted in Mirko Perardini's lab, both focusing on immune-based strategies for an HIV cure, and both illustrating how non-human primate studies can help move this field forward. So the first study looks at venetoclax, a molecule that can promote the death of infected cells. And the second study explores a combination of 2 inhibitors aimed at boosting the immune system to better fight the virus. So in this introduction, I will briefly revisit some points that Afam already covered in the previous presentation, but it's always better to reinforce key ideas, right? So I think we are all familiar with this first slide, but I will give you a quick reminder. So early after infection, the virus in the blood called viremia rapidly increases, and at the same time, the latent reservoir where the virus is hidden is established. But where does HIV hide? Actually, HIV hides in different immune cell types, but mainly in CD4 T cells. And those immune cell types are themselves located across multiple tissues, such as blood, lymph node, spleen, the gut, the brain, or the liver. But now, antiretroviral therapy ART can suppress viral replication, but it fails to eliminate this latent reservoir, which is actually the major obstacle to HIV eradication. And because of this viral reservoir, if treatment is interrupted, the virus quickly rebounds and control is lost due to an impaired immune response. So the key challenge is actually how to eliminate these reservoirs within a very complex system involving viral persistence and immune dysfunction. So to study this in more detail, we rely on non-human primate or monkey models such as rhesus macaques. As Afam previously explained, in rhesus macaques infected with HIV, so the similar virus of HIV in monkeys, the overall viral dynamics are very similar to what we see in humans. We see a rapid increase in viremia, an establishment of the reservoir, viral suppression under ART, and a rebound after treatment interruption. And because of this very strong similarity in viral pathogenesis, this model is highly relevant and essential for HIV core research, because this model allows us to have a precise control of viral dose and timing of infection and treatment. It also enables rigorous evaluation and comparison of different interventions. And more importantly, it gives us access to both blood and tissue compartments, including key sites where the virus persists, but also where immune responses take place. And more broadly, NHP monkey models are essential across the field, from pathogenesis, so how the virus causes the disease, to prevention, transmission, vaccines, treatment, or cure strategies. And today, let's talk about, I will focus actually on these two applications, targeting the viral reservoir and improving the viral control after the therapeutic interventions. There are actually several time points where we can act. We can act early at ART initiation to limit the establishment of the reservoir. We can also act during long-term ART where people are fully suppressed, are undetectable, to try to reduce the viral reservoir. But we can also act around treatment interruption here or here to delay or to control the viral rebound. So I'm going to start with this first strategy by showing you an example from a study we conducted in the lab where we act early at the time of ART initiation to limit the establishment of the reservoir. So one approach we are exploring is based on inducing the cell death of infected cells. This work was led by Thomas Salinas in the lab and focuses on targeting a protein called BCL2. What is BCL2? Actually, BCL2 is a protein which helps cells survive. So by inhibiting this protein with a molecule called venetoclax, we aim to selectively push infected cells to die. So the idea is that in combination with antiretroviral therapy, this could help reduce the size of the latent reservoir. So to understand this approach, let me briefly explain you the role of BCL2. So under normal condition, when BCL2 is normally expressed, infected CD40 cells can be recognized by the immune system and eliminated. Or the infected CD40 cells can also die by themselves. However, when BCL2 is overexpressed, and that happens during HIV infection, infected cells become resistant to cell death. Why? Because they are not efficiently eliminated by the immune system, and they are not able to die by themselves. So the HIV can persist in the body. So the goal is to block BCL2, the bad guy, in order to restore the ability of infected cells to undergo cell death. For that, we used venetoclax. And the advantage of venetoclax is that it's already FDA approved and used in cancer to promote the death of cancer cells. So based on this rationale, our hypothesis is that combining BCL2 inhibition with early ART could limit both the establishment and the maintenance of the viral reservoir. So in our lab, we used 3 groups of animals. One group with ART alone, one group with ART plus venetoclax, and another group treated with ART and venetoclax, but also where we depleted CD8 T cells. So CD8 T cells are cells capable to kill infected cells. And we used this group to better understand the role of the immune responses. But I won't go into detail on that today at all. For now, just keep in mind that both group 2 and group 3 received venetoclax. So venetoclax was given during early ART, as you can see here in blue arrow, and we followed the animal over time, collecting samples from blood and lymph node. And collecting blood and lymph node allowed us to assess both immunological impact and the effect of on the viral reservoir. So with this study, we asked 2 main questions. First, does our intervention have an impact on the immune system? Second, does it change the size of the viral reservoir? So let's start with the first question. We looked at the immune impact of our intervention. What we see here in blue is that CD40 cells, the main target cells of the virus, are significantly reduced in animals receiving venetoclax here in blue compared to the animals that did not receive venetoclax in black. And importantly, over time, as you can see here, CD40 cells levels recover despite this initial drop, which is actually reassuring. So overall, this confirms that venetoclax is active and has the expected biological effect. Then, to go a bit deeper, we looked at gene expression in CD40 cells. What does that mean? It means looking at which genes are turned on or turned off in their cells to understand how they are behaving. And here we can clearly see in red an increase in genes associated with cell death in animals receiving venetoclax compared to controls. So overall, this shows that CD40 cells are more prone to die under venetoclax treatment, which is exactly what we expect from BCL2 inhibition. So this is a good news. Now, let's look at whether our intervention affects the size of the viral reservoir. First, just to explain how we measure the viral reservoir. We isolated blood cells. We then purified CD40 cells, so the target cells of the virus, and then we used a technique called IPDA to quantify the number of infected cells. And then, using this approach, what we see is a significant reduction in viral reservoir in animals receiving venetoclax, as you can see in blue compared to control in black. So this suggests that a preferential elimination in infected cells, and importantly, this effect is rapid and sustained over time. And I also don't show here, but we observed a similar reduction in lymphoid tissues. So this indicates that blocking BCL2 leads to a lasting reduction of the viral reservoir. So to summarize, we tested BCL2 inhibition using venetoclax at the time of ART initiation, and we found that this approach rapidly and significantly reduces the viral reservoir, both in blood and lymph node. Importantly, this reduction is maintained over time. And also, venetoclax appears to preferentially target infected cells. And another good news is that this work is currently available as a preprint online and is under review. And more importantly, this work has direct clinical relevance. Why? Because there is already an ongoing clinical trial using venetoclax in people living with HIV on ART, led by Thomas Rasmussen, with very promising safety data so far. And based on our NHP study design, a new clinical trial just has been approved to test venetoclax at the time of ART initiation. So this really highlights how findings from non-human primate studies, from monkey studies, can directly inform and accelerate human clinical research. And beyond HIV, this approach may also have relevance for other infections, such as tuberculosis. Actually, the bacteria responsible for tuberculosis can induce BCL2 to prevent infected cells from dying, which helps the infection to persist. So by inhibiting BCL2, we can promote the death of infected cells, improve bacterial clearance, and potentially reduce tissue damage. So again, this suggests that targeting BCL2 could be a broader host-directed strategy against persistent infections. Now, moving to the second part of the presentation. Here, instead of acting early, we focus on strategies during ART and around ART interruption, here to improve viral control and to delay the rebound, as shown by the red arrow. In this second approach, instead of targeting infected CD40 cells directly, we aim to empower the immune system. The idea is to block what we call immune brakes mechanisms that normally limit immune responses, but that HIV can exploit. And one of these is TGF-beta. TGF-beta is the bad guy. TGF-beta helps the virus hide by suppressing the cell's natural antiviral defenses. Also, TGF-beta increases another molecule called PD-1. PD-1 makes immune cells less active, more dysfunctional, and importantly, more likely to harbor hidden virus. So together, TGF-beta and PD-1 prevent the immune system from efficiently clearing infected cells, allowing the virus to persist. So our hypothesis is that blocking both TGF-beta and PD-1, we can restore the immune function and improve the ability to control or eliminate the virus. So based on this hypothesis, we designed this study. So we used 3 groups of animals, a control group receiving ART only, another group receiving a protein that blocks TGF-beta called anti-TGF-beta, and a third group receiving both anti-TGF-beta and anti-PD-1. So all animals were infected by HIV and placed on ART. Then during long-term ART, when animals were fully suppressed, so fully undetectable, we administered the treatment every 3 weeks before and after the ART interruption. And then we followed the animals over time, collecting samples from blood, lymph node, the gut, bone marrow, to assess immune responses and viral outcomes. Also, importantly, the animals were followed for 6 months post-ART interruption. So for this study, we asked 4 main questions. First, is the intervention safe? Second, do we reach adequate drug levels in the blood? Third, does it impact the viral rebound? And finally, does it have an effect on the immune system? First, is our intervention safe? Actually, we did not observe any harmful effects on blood cells, and the routine lab test showed no concerning changes in kidney, liver, or heart function. We observed 2 adverse events. So 1 case of skin irritation that was easily resolved with topical care, and 1 animal whose immune system reacted against the treatment. So this animal was removed from the study. But overall, the combination of anti-TGF-beta and anti-PD-1 is well tolerated in rhesus macaques. So next, we wanted to make sure that the animals actually reach adequate levels of the treatment in the blood. So here we measured the levels of the drugs in the blood over time, and as expected, in the treated animals, we clearly detected anti-TGF-beta, as you can see on this top panel, and we also clearly detected anti-PD-1 on the bottom panel. So this confirms that the animals are reaching adequate levels of both treatment. So next, and the next important question is actually, can this intervention actually delay or control viral rebound? And here we are looking at viral rebound after stopping ART. So in the control group in green, most animals rapidly rebound and only about a third shows a controller profile. And with TGF-beta blockade alone, we don't see much improvement, very similar to the control group. But when we combine anti-TGF-beta and anti-PD-1, the picture changes dramatically because about 83% of the animals are able to control the virus even several months after stopping ART. So this suggests that the combination therapy significantly improves the ability to control the viral rebound after ART interruption. Finally, we asked whether this intervention also has an impact on the immune system. So we looked more closely at how the immune system is responding, and on the left, you can see the overall CD4 T cell counts over time. So after ART interruption, animals receiving the combination treatment in blue show a better recovery of CD4 T cells compared to the 2 other groups. And on the right, we focus on a specific subset called effector memory CD4 T cells, which are important for immune responses. And here we see a clear and sustained increase in their cells in the combo treated animals. So this suggests that the treatment not only helps control the virus, but also support the recovery and function of the immune system. So to summarize, what we did, we tested a new cure strategy in a non-human primate model, combining 2 treatments that release immune breaks. And we asked whether this could help control the virus after stopping the treatment. And we found that this approach is safe with no major toxicity observed. We also see a promising effect with most treated animals showing better control of the virus after antiretroviral therapy interruption. And importantly, we observed immune improvement with recovery of CD4 T cells and better maturation of their cells. Finally, why this matters is that similar approaches targeting PD-1 are already used in patients, for example, in cancer, and targeting TGF-beta is an emerging strategy. So together, these findings bring us closer to the goal of long-term HIV control without the need for daily treatment. So to finish, this is the end of my presentation. And to finish, I would like to acknowledge everyone involved in this project. This includes my colleagues in our lab and our collaborators at Emory, but also external collaborators. I also want to thank the veterinary and the research service teams who do incredible work here and make the studies possible. We are also grateful to Merck for providing the anti-TGF-beta and anti-PD-1, as well as Gilead, ViiV for their support. And of course, I always like to thank the animals without whom this work would not have been possible. And thank you for your attention.
Terrific. Thank you so much, Julien. That was great. So everyone, please, if you have questions, you can either put them in the chat or raise your hand. I'll start off with one question while people are getting their questions queued up. So that last study was really fascinating with the anti-TGF and PD-1. So as a result of that, well, has it been published yet? And is that leading to trials in humans yet? Do you know?
So no, not for this study.
Okay.
No, not yet. Anti-PD-1 is already used, as I said, but anti-TGF-beta in humans, we don't have this kind of anti-TGF-beta in humans yet. But this is very promising for future clinical trial, since we see, this control profile in 83% of animals.
Right. And then Michael had a kind of a follow-on question to that. What specific decision will this study inform? Go, no go, dose, safety, those kind of questions?
So the next step is actually to prolong the post-ATI phase and also to do more doses of anti-TGF-beta and anti-PD-1 to see if adding more doses will improve the viral control. And then we could follow maybe not 6 months, but 1 year or 2 years or 3 years. So we need to push more the studies forward.
Okay, great. Thank you. Now, the next question I see is from Liz Heileman. Liz, go ahead.
Hi, thank you. I'm a medical journalist who's following this sort of debate about animal testing quite closely. And I think this presentation is missing the point a little bit. I mean, when people are debating animal research, that they're concerned about the welfare of the animals. And I know there's a lot of safeguards in place, but, people will show an image of, a monkey or a beagle in cages or being restrained for testing. And that has an emotional effect on people. And it's not so much that people are like, what specific things are these studies teaching us, but, are they the right thing to do? And I think, that needs to be addressed, because when the arguments about funding and stuff come up, that's what the people who are opposed to it care about.
Yeah, this is a very, very important point, and one that we take extremely seriously in our daily work. Beyond procedures, beyond interventions, animals are housed in environment with social interactions to support their well-being. When they are assigned to a study, they are paired. They are daily monitored by the veterinary, by the technicians to ensure their well-being, to ensure their welfare. They are not, and also there is an independent federal committee called Institutional Animal Care and Use Committee, YACAC, which ensures that every procedure is justified, that no alternative are available, and that animal welfare is fully considered. So everything is extremely strictly how do you say in English?
Regulated.
Regulated.
Yeah, yeah.
So, but I understand the fear of people, but everything is considered. We are not doing that in an evil way. To minimize pain, discomfort, we use the same principles as in human medicine. For example, when we have to do a blood draw, we do a short anesthesia. And when we have to do a biopsy, we do a longer anesthesia. We use pain relief. We use analgesic, anti-inflammatory drugs. Everything is regulated.
And just to highlight, in addition to what Julien says, I mean, I think that, to be honest, we probably have not done a good job as also, I mean, we just assume that people trust the scientists are doing responsible research, and we're really not just out in to torture monkeys. But unfortunately, the voice in the room got really loud from the other side, where I think a lot of it has been misrepresentation of what animal and primate research in particular is really about. And, I think now, as a field and as a community, we're probably really all galvanized now to make sure that we are we can explain what we do, and the idea that this type of work is really highly regulated. We do it in a humane way, and the animals, as much as possible, we make sure that we get cared for. I mean, the health of these animals, the veterinary care is exceptional. I mean, much better than what some people are getting, and to be honest, because they have people assigned to them. They are watched every time. There are regulations in place to make sure that animals are not subjected to unnecessary pain. So I think your point is well taken, and I think as a field we're really committed to ensuring going forward that we also make our voices clear that then the misrepresentation is not true, and that we're doing things in a way that is really humane. But they're important and they're essential because we don't want to be harming people.
Thank you for that, Linda. You had your hand up.
Yeah, you know what? Well, let Michael go first because his is
Okay.
more on topic. Mine was more science related to this, to venetoclax.
Go ahead, Michael.
Quotes or a lot of questions in the chat. But one of the questions I had is, how do you engage community stakeholders in the shaping and the reviewing of this NHP research? Or do you have community stakeholders involved in that part? We're used to it in terms of human trials. I didn't know if there's any sort of community stakeholder work in NHP trials specific to that. Do you guys have any sort of community stakeholder mechanisms?
I mean, we're part of the Delaney, and Delaney has a CAB, right? And so we always present our work to a CAB and really to get input. I mean, I think it's really important for us as we do studies in non-human primates to consider what the community is interested in, how the community thinks our treatments might actually be translatable, and so it's really important for us to get that feedback from the community. As I said, most of our studies are regulated by ICO. We have to get approvals to even do them. But even when we do them, I think it's really critical for us. And for us through the Delaney collaboration, I think it's the same as Erase and all the other collaboratories. The idea was that we would come to our meetings and highlight the work we're doing and actually get feedback from our CAB to see what they think about us. And actually, interesting, like the Keystone I just was at, I had with interesting conversations. We had a panel with the NIH, and some of it was about implementation science. And one of the questions we were asking them is like, there's a new push from the NIH about how to implement and moving towards implementation science. And we wanted to know, those of us who do pure research and basic research, and we don't have tools yet to implement, what are they thinking for us? And I think for them, going to your question, they want us to be very considerate in terms of when we're developing and testing new interventions. How is this going to be translational? How are we going to implement these new interventions, starting from the discovery phase? And I think so. I think those conversations are going to start increasing more and more as we go forward in this next season.
And I also had a question about ATI studies, the analytical treatment interruption studies. Do they use NHP in those type of studies? Do we put animals under treatment interruption like that? And if we do, how does that model the human ATI experience?
Absolutely. Okay, go ahead, Julien.
But can you repeat the question? I didn't hear you very well. Actually.
Basically, I was just wondering, like, do you use NHPs for treatment interruption studies, the analytic treatment interruptions that we're doing in humans, and how closely does the animal model the human ATI experience? You know what I'm asking? Just like, are we using NHPs in ATI studies? If we are, how close is the model modeling the human experience?
So I think the answer is very simple, because the viral dynamic at interruption between animals and humans are extremely similar as well. So it's very translational. That's why. And also Chinese and Indian rhesus macaques are very reliable for that. So that's why we are also using several groups of animals to compare all the different interventions and everything.
Great, thank you. And also, I just know that in terms of NHP studies, there's these 3 R's concept, the reduction, replacement, and refinement. And I just wanted to bring that up because I don't know if people are familiar with it on the call here. But could you briefly explain that concept of what the 3 R's are about?
Yeah, the 3 R's are 3 terms: replacement, reduction, and refinement. So when we talk about replacement, we want to avoid or replace the use of animals wherever possible. Reduction is to use the fewest animals necessary to achieve reliable results. And then the last is refinement. So we want to minimize, when we use animal, we want to minimize the pain and suffering and distress to improve the animal welfare. In NHP study, in HIV cure research, we are doing that too. We are applying that. However, replacement, complete replacement, as explained, Afam, is not possible in the HIV cure research, because we need this biological complexity to assess and to address the biological, the complex problematics that has HIV cure. We need a whole organism within the whole, immune interaction system. So, but we are applying the 2 other R for HIV cure research.
Awesome, thank you.
Thank you
Michael,
for that,
you
Linda.
want to go through the chat and do that and get the questions?
Yeah, that's what I've been doing. Most of them are from me, but I wanted to turn it over if there's other questions that people have, just to give people a chance.
Let me ask Julien my question. Julien, when you were talking about venetoclax, you said that the intervention was maintain control over time. Can you tell us how much time you were talking about there?
I think that Thomas did 1 year.
Okay, so that's as far as he is.
I'm going back. Days 284. So not 1 year, but almost.
Yeah. And I just have to say, I mean, certainly we understand that you need control arms, and we're in the beginning of cure research, and we need to prove what drug does what to who, and that's all good and well. But you can also compare arms, and see what happens. And it's extremely important, I think, what you all have, the design that you have or going forward with here is using the antivirals as well to see what happens, because it feels to me, and it's just a feeling that I believe needs to be studied, that we need to study these new interventions with antivirals. I mean, maybe, you're talking about 2 different things. You're talking about live active virus and virus that is not always dormant, but sometimes dormant, and then blows up. And so we may be talking about 2 different things, so we may need 2 different interventions there to deal with that. So I mean, in order to give the intervention a chance, you might have to be able to control the virus that's already there, alive and well and dancing and singing, tap dancing. So I just want to put in a plug for continuing to do that and thinking in those terms, rather than trying these interventions, the same sorts of things together, or we need combos of things. I think you get what I'm saying without further.
Yeah, I get the point. Yes, ideally it would be ideal to have 10 arms to have all the controls as possible.
Yeah.
But what we need is the money to do all these
Right.
arms.
Well, 10 arms just goes a little crazy too, because then
I know.
you don't know anything when you get done. With 3 arms is great. Or 3,
Yeah.
it's really we've done that in the past. We made history by changing research to make researchers do things that way to save time and to be more helpful to participants and eventually patients. But, I'm just saying yay to that.
Yeah.
Okay, thank you. Liz has her hand up. Go ahead, Liz.
Sorry to speak again, but following up on the replacement point that was just made, what kinds of things do you think could potentially replace animal studies? I just put something in the chat about an article I just saw about using lymphoid organs, perhaps. Is that something that is being worked on and shows any promise?
Afam, you want to answer? I can answer that too if you want.
Yeah, I mean, there are different organoid models, and there's a huge investment now by the NIH to develop more and more of these human-based organoid systems. And those organoids are like in vitro cultures that's supposed to a certain degree, mimic what a tissue system should look like. So basically, lymph nodes in particular are sites where if an individual has exposure to a new pathogen, like an infectious agent, the immune cells take parts of that virus or that agent and it traffics to the lymph nodes. And that's in the lymph nodes is where the immune system now begins to get educated on how to fight that pathogen and make antibodies. And those antibodies are produced and then the system goes out of the system. So lymph nodes are very important for the immune system. Now you can make organoids, that's these in vitro cultures that look like and operate like these in vitro systems. So there are new organoids being developed. They're organoids for brains, they're organoids for lymph nodes, there's even gut organoids that are being developed. And a lot of these are very in early stage. And I think a lot of them are very encouraging because they are human-based ones. So as I said, it's challenging to access these tissues in people all the time. I mean, people don't always want to be subject to getting a tissue biopsy if they don't need to. But it's the only way we can study it. So there are many of these organoids that are being developed. A lot of them are very early stages. And I think a lot of them are very good for specific questions. But they're not for all questions, because as I said, if you have an infection in your gut and you want to see how the T cells you've educated in the lymph nodes traffic to the gut to go and take care of the infection, you can't do it in the in vitro system, right? So it's all about the question you're trying to answer. If the question is right, you can use the organoids to answer that question. They can be very potential for brain organoids, for instance, if you're trying to understand how HIV persists in the brain or how an intervention or treatment might affect HIV in the brain. Because it's a closed system, if you have a brain organoid and that brain organoid is infected with HIV, you can introduce a drug or a treatment and look at toxicity, look at synapses, signaling, all these things you can do in the brain that's hard to do in people. But if you're looking at it in terms of a system-wide level, how this intervention impacts the whole system, then it's challenging to do. So it's all about the question you're trying to answer, and how and the interpretation of that question. And hopefully over time, we will get to systems that can actually be validated, that we no longer need to answer those questions in vivo. But right now, I think we're not there.
Yeah, thank you for mentioning the organized.
This is the important thing. It's complementary, but as said, Afam, we are not there. But all the tools are extremely important for the future because we are improving. But right now, we can replace. We really need to understand how a drug behaves across the whole body, and including the interactions between organs or with the immune system.
Thank you for that. I just wanted to piggyback on what Afam was saying about the organoid models. Many of us know about the last gift study, which is in people living with HIV at end of life who are dying, who agree to a rapid autopsy. And so one of their next steps is going to be looking at using those tissues from the brain and other reservoirs that are organs that are hard to access during life to create those kind of organized systems. So it's definitely where the field is going.
That's really fantastic. But just a point to say about the last gift cohort, just as people don't know about it, this is this amazing cohort of people who have gifted their bodies to HIV. Just to highlight the fact that This is happening in people who are at end-stage disease for other things, especially chronic end-stage disease. And so while it's going to be a fantastic model, we also have to be very careful because it doesn't necessarily, their systems are at an end stage of disease that might not necessarily reflect what's happening in people who are exposed to HIV when they're infants or in adults or young people who are relatively healthy. So these are all complex and challenging, but it's really an amazing tool that we have in the field.
Another question I had was, I've heard, and correct me if this is not right, that often the animals used in experiments are almost exclusively male because they're keeping the females in the colonies for breeding. Because as you mentioned earlier, they, it's kind of a self-sustaining population. Is that the case? And do they even look at gender in the animal models that they're using?
Julien, did you want to answer that? Or I can if I want, but
yeah, it's true that, for example, in my study, most of them are male, but overall, in my previous study, in my experience, when we use female and male, I did not observe significant differences between genders. Yes. But of course, there are some kind of differences due to the genes Y of the female in terms of immune response, but not that significant.
And studies we're doing, we have access to females, and we are using females. The challenge is we can't have, because of the cost, we don't have that many animals within a group that are either male or female for us to say, is there a difference? Is there a sex-based difference? There are specific studies that are specifically male only or female only because we want to ask those questions in the context of the sex of the animal. But to look at differences across sex is a challenge just because it really requires a lot of animals. And these experiments are already challenging anyway. So sometimes you want to control so many variables so that you can get a specific answer. You have too many variables, then it might be challenging to get us an answer just because of the animal sizes we often use in these studies.
Thank you. Michael, I see you had some questions in the chat. Go ahead.
Oh, I was just wondering, given the idea of limited resources, is there any tension between cure work versus prevention work or treatment work? In other words, is there tensions between
we lost you.
We lost you for a minute there, Michael.
I think he's saying, we see that in real life. Prevention.
Oh, well, they're using the money for this.
Oh, yeah, there you go. Do you?
I'm going to turn off your video in case it's a bandwidth issue.
But I think I got the crux of his question. Is there a tension between prevention and access to resources to these experiments? I don't think there is tension. I think a lot of the studies we do are very complementary. Sometimes I think it's very informative because some of the things they're developing in the prevention space with vaccines and these new methods to generate potent antibodies, there are also things we're interested in cure. So I think a lot of it is very complementary, understanding how these different agents interact with the immune system and how the differences or similarities with what happens when animals are on therapy versus when they're just not infected at all. I think that these things help inform each other. So I don't see that there's a lot of tension or resources. I mean, obviously resources are really at the funding level, but the NIH I know is really keen and interested and is really wants to continue to promote HIV cure research across the spectrum, both in vitro animal models as well as people-based studies. So I think so far the funding is not so excessive that we have a limited resource. I think it's all about funding priorities.
I think the problem in human beings is that things are siloed in the government as far as prevention is over here and then treatment and then, research. And they're all rivals for the same pot of money when you are looking at animals all at the same time and dealing with all of those issues at the same time rather than, okay, here, 3 different pots. You got one pot and maybe you don't have one pot, but, it's a little different.
I think the challenge now in the prevention space is how to deal with the impact that long-acting antivirals are going to make on the prevention field, and the role of a preventative vaccine in that space going forward. I think that is where the challenge is. But I don't think there's a question for cure right now, because we don't have any tools other than, the remarkable progress that we've had with Mark and all the other wonderful people who have been cured for HIV so far. That is not something as a tool that we can implement. But when we have more and more tools, then that's what we were hoping to have, more and more tools, more and more options for people in the cure space as well. And I think the non-human primates are going to have a big role in developing those new tools that they have in prevention. They have many tools now in prevention, but we don't have those tools yet for cure.
Is that all the questions?
There was one that just came in right now. It's a long question. So take a look at it.
I don't see
From
it.
Paul. It's just the very last one.
I see it.
Yeah, I didn't mean
Paul,
for
go ahead, Paul.
didn't mean for it to be so long, but
Make it
took me a
short,
while
Paul. We
to think
got
it through.
3 minutes, and I need a minute at the end. Hi, by the way. Nice to see you.
Good to see you too. Thanks, Linda.
Okay. So if I recall, this is about using the ethics of using animals for research in general, basically, and because they are biologically similar to humans. Is that the summary of your question?
Right. I mean, I think the results are very important, and I appreciate the idea that speed is of the essence, but I still get hung up on the idea that we are another species in the world, and somehow just assuming we have the right to take on another species and exploit them.
Yeah, I mean, I understand your perspective and I respect it. But I also think that for medical progress, and these studies are important. And as I said, our goal is to do our best to find ways to find alternatives to these. And when we do find these alternatives, we shift, because we're not going to be given the opportunity to do them when there's an alternative available. I mean, even for us to apply for funding to do these studies, we will only get funding if we can convince the funding agencies that this is the only way we can answer this question. And I think over the years, and now that there's more investment by the NIH to actually find alternative to it, you will see a natural decline over time, but we have to put the work in. And I think that's what the field is moving towards. We have to validate that these alternatives are better. And once we validate these, then we will be using them instead of doing animal work. So I see your point, and I think the field is working towards that, but we're not there yet. And there's still questions that we cannot just, especially pediatric and things that go into infants and children. It's really, really hard to just take something you've developed in vitro and put that into a healthy child, or to say that this might work in children, even in adults. Even what we're doing in cure, it's really challenging to do these studies and understand if mechanisms are going to work. And we always want to develop treatments that are not going to do harm in people. So I see your point, and I said as a field, we're working towards the same goal. We want to reduce the use of animals. I mean, it's very expensive too. It's a huge investment to do this type of research, and we only want to do that when alternatives aren't available.
So, I think that the purpose of this call today was to talk about all of these different perspectives, and to hear what you all had to say about why we need this, what we're doing to make animals more safe and comfortable, and Paul's perspective about why are we doing this at all, Liv's about other different ways to do it. So I think that we achieved our goal today. I think we learned things, and we were respectful of what other people thought too. So thank you. Go ahead, Afam.
There was one question about females that maybe I just wanted to highlight. We are using females, and we have studies that are female only. So we aren't excluding females from any of our studies. I just wanted to highlight that we are. It's just because of when I was referring to females, it's about looking at sex-based different, and does this affect male and females differently? It's hard to do that in every study because the numbers are not there. But we do include females in our studies. We try and actually, right now, our group, we try and balance our groups so we have an equal number of males and females within our studies to ask the same question. But we don't exclude females at all from our research. And we're interested in actually now looking at how HIV interacts at women as animals age. Animals get female cascade menopause, the same as is in humans. And we're trying to understand reproductive health and all these other things that happens with when animals are infected with SIV. So just to kind of highlight that.
Great, thank
Well,
you.
okay, so thank you, speakers, Afam and Julien. Thanks everybody for your wonderful questions and for participating. We're really glad that you all participated today. And don't forget, May the 7th, same time, but on Thursday, May the 7th, we have Nadia Rowan that's going to talk about block and lock, a different strategy for controlling the reservoir, number one, and how it relates to aging and metformin, which it's unbelievably promising. I hate to say that because it's not like me to push something that's not tested for, A to Z, but it really does have, it looks like it has potential. So I'm not saying it does, but I think it's extremely interesting, and you'll be interested to learn what Nadia has to say. So, anybody else? Final thoughts, Michael?
Nope.
Jeff?
Nope, terrific. Thank you so much to our speakers and everyone for joining.
Thank you.
Okay.
Thank you
All
for
right.
having us. This was fantastic. Thank you.
Bye-bye. Thank you.
Bye.